Is it okay? All right, so we ended by talking about bacterial fitness. This is going to be a talk about speaker fitness. Can I do this for a second hour? So now we're asking the question, can we design new function in proteins? And I left you with this sort of question in the middle of my talk before lunch. Given that we've been successful robustly and reproducibly in designing folds, sequences that fold into a unique conformation designed on the computer, what would you expect? Is design a function going to be more, is it going to be easier or harder than design of folds or structures? And when I first saw as a graduate student the top 7 paper, I had no doubt in my mind that actually this is it is a remarkable achievement, and that designing function, which is what I've been interested more than design of folds, is actually going to be easier. And the reason for this is that whereas in fold design, you're designing, in the case of top 7, 93 residues to fold into a predetermined conformation, given all the huge space of alternative conformations, and you're able to do this, again, reproducibly. Design of function, at least in our minds, seems to be an easier problem, right? You have thousands of proteins which you can take molecular structures of from the PDB, and all you need to do is design an active site. Right? An active site can be between, let's say, 8 and maybe 15, maybe even 20 amino acid residues on the surface of the protein. You know that the scaffold is going to be stable, right? You're taking it from a natural protein. It should be stable. So what's the problem? Designing 10 to 20 amino acid surface amino acid residues sounds like a much simpler problem. This turns out not to be the case, but I've spent the last 10 years first thinking that it is the case, that the same methodology that gave us top 7, essentially where you drill down the energy landscape, finding deeper and deeper minima by this conformation and sequence search, that this strategy alone will give us binders and maybe enzymes in the future. But so first I tried that strategy. When it failed, I did something else, and I'll show you. This was during my postdoc with David Baker. And since then, I've realized that this strategy actually doesn't actually work, given our current understanding of the energy function. I'll show you why, and then I'll show you the strategies that we've been developing in our lab here to overcome these problems. Come on. All right. So as an outline, current successes and challenges in design of function. Again, I'm just going to talk, just giving you a flavor, not comprehensive view of the field. What we've been developing is antibody and enzyme design. And last, I'll show you something that was sort of like a side effect of a project on antibodies and enzymes, where in order to design new function in antibodies and enzymes, we first needed to understand how to design, again, stable proteins, stable folds. And as we did that, we learned some lessons which we can actually implement for designing very tough to work with proteins, and we've had some success with that, and I'll show you that as well. So to start, this is from my postdoctoral project with David Baker. It's all been published, not this publication. It's all been published already. I'll just give you an overview of this work. During my postdoc, I was interested in the question of how do you design a protein that would bind specifically to a target site on a protein you're interested in. And our model system for this was hemagglutinin. This is the same hemagglutinin that Stephen Harrison mentioned 2 days ago. This is the protein involved in membrane fusion between influenza virus and human membranes. And if you recall what Stephen Harrison mentioned, there's this stalk region at the bottom near the viral membrane, which is responsible for the fusion event. There's a huge conformational change. This region participates in that huge conformational change. And we know already from work from Ian Wilson's lab that if you stabilize this state, for instance, if you use an antibody to bind at that state, in this conformation, you can stabilize the pre-fusion state of hemagglutinin and prevent what Stephen Harrison mentioned as this catalysis, the ability of hemagglutinin to catalyze the fusion of the viral and host membranes. So antibodies are great. but in this case, they're not so good as potential applications and therapies against influenza, because influenza virus actually evades antibodies. Antibodies are large, and as Stephen mentioned, the viral envelope is very dense with hemagglutinin molecules. Antibodies have a hard time diffusing down there. We thought that if we could design a small protein that would still bind in this site, unrelated to the antibody, of course, we'd have a better chance of stabilizing the prefusion state and serving as potentially even a therapeutic. So our strategy begins, again, we had to give up this idea of just drilling down the energy landscape. Our strategy begins by saying, okay, this is the part of the surface that we want to bind on the stalk region of hemagglutinin. We start the process by docking individual dismembered amino acid residues, sort of like what you would expect from small molecule drug design. You just dock the amino acid residues against the surface and isolate constellations of residues, different constellations of residues that bind favorably, that are predicted to bind favorably to the target site. And you do this many, many times. You have dozens, actually. If you do the combinatorics, it's thousands and thousands of different constellations of residues. For instance, methionine here, phenylalanine, and tyrosine here. And then after you're done with this, you go and scan the protein data bank for protein structures, again from nature, that could present this very small site, we call it the hotspot site for interaction, present this site and stabilize it, and then form additional interactions. 3 residues is not enough to interact with high affinity with any target protein. So that's generally the strategy. You can see here already that we're not just asking for the minimum energy sequence conformation. We're asking for something a little bit more complex. It's a protein that would present these particular residues in particular conformation so that it binds in this particular mode to influenza hemagglutinin. And these are just 2 of the proteins we designed using this strategy. These, the scaffolds on which these proteins are based are completely unrelated to the immune system. They actually come from other life forms. This is a 96 residue protein from a hyperthermophilic protein of unknown function. This is a fragment. The scaffold is a fragment from a plant transcription factor, completely unrelated even to protein binding. And yet, after our design process, this you can see here the 3 residues of the hotspot here, just 2 of the hotspot residues, and design of the surface. It's not just these 2 or 3 residues. It's about 15 residues on average for each scaffold. These 2 proteins actually bound, initially at low affinity, and then through affinity maturation at high affinity to the stem region. How do we know that? Because Ian Wilson crystallized the 2 proteins with Spanish flu hemagglutinin. And what you can see here in gold is Ian Wilson's structure. And you can see that virtually every heavy atom at the interface is right where we designed it to be. And this is the first demonstration for atomic accuracy design, de novo design of function. The proteins, again, after affinity maturation, actually bind quite tightly with subnanomolar affinities to hemagglutinin. They're better in that sense than many of the antibodies that are being developed as potential therapeutics. And they also bind more broadly to different influenza strains than natural antibodies. So this provides some sense that maybe there's a beginnings for an application directly from computational protein design, maybe all the way even to a therapeutic, although these proteins, again, are so out there. They're derived from bacterial proteins. We don't know how something like that would react with a human body, although we know that in animals they do protect against influenza infection. Okay, any question about this? Okay, we've been able to apply the same strategy to other target proteins. Here you see again the flu binders. This is a binder of IgG1, the Fc domain of IgG1 antibodies, and here is an enzyme inhibitor targeting lysozyme. So each of these is a different target, very different molecular features, and yet we were able in each one to design binders. So this seems like it's quite a successful methodology. But if you look at these examples, what you'll see is that in all of these cases, the binders have very high secondary structure content, right? Either helices or sheets at the interface, almost no loops. There's this very short loop here.
What do you call affinity maturation? This is the mutation selection?
Yes, it's what we did is either site saturation mutagenesis, as I showed for membrane proteins, or error-prone PCR, and then selection through yeast display, and binding, more and more stringent binding affinity.
So in what sense is your strategy different than regular?
Finding the original,
So
the initial.
because you start with a more restricted initial.
Much more restricted. But also it's targeted. We can say, so if, in the case of hemagglutinin is a large molecule. You can get our own bodies produce many antibodies against hemagglutinin. But if you look at these antibodies and the molecular details of how they bind hemagglutinin, it's almost always at the top of the molecule, almost never at the stem region. And that tells you that if you don't know how to direct the antibody selection process, you won't be able to achieve what you want. And these binders, since they were computationally designed to bind the specific site, you can do that. Yes?
So when we talk about proteins, you said that proteins of initial designs are very stable. Why are binders very unstable in the design, not also very stable like you were?
Yes. Okay, great. I'll get to this point exactly. Yes?
If I remember correctly from Stephen Harrison's lecture, the fusion happens in an intracellular visit.
Yes, in the zone.
What's the therapeutic intuition? How do you target hemagglutinin when it's already in visit?
What they do is just essentially as a prophylactic. With these animals, you give it to the lung, to the airways. Then when
Have it doesn't work.
pardon?
Once you're infected.
It can, well, the infection process is an iterative process, right? It's an exponential process whereby viruses produce in one cell, 1 million variants are produced in one cell. They then bud out and infect other cells. If you can intervene at that step, you can stop the exponential
So you
phase.
have to introduce prophylaxis. By the time you have infection, you notice that infection, you can't treat it.
No, it can be treated, right? Because infection is a continuous process. Once it ends, it ends. But I don't want to highlight. I really don't want to emphasize the potential applicative of such molecules because we really don't know. These are synthetic molecules taken from very different sources than what we're used to in generating medicines. I don't know whether this will work. It's more a question of fundamental science. Can we design proteins that bind actually a challenging target site? Okay? So we haven't been able, even though we tried, we haven't been able to design function in loops. And this is a really significant constraint on our abilities to design function. Because if you look at natural proteins, which are involved in the prototypical proteins that are involved in function, be they antibodies for binding or timbrels for catalysis, such as this timbrel shown here, what you see is that there are secondary structural elements, but often the molecular recognition components are situated on loop regions, as you see here. Here, essentially, almost all the catalytically important residues are situated on loop regions. And the reason is that these loop regions are much more versatile, right? You can insert and delete residues from loop regions. You can't do this. Normally, you cannot do this to a helix or a sheet. So nature has been taking advantage of this plasticity of loop regions in order to encode new functions or to expand the functional repertoire of protein families. If we're unable to design function in loops, that's a significant constraint on our ability to design function. And this has been sort of the leading or the guiding strategy in or rationale for our work, even though it's very hard to design function in loops, and we'll talk about why that is in a second. We really have to do that in order to move on to the next stage. So why is it so hard to design function? Why has it been so easy to design protein folds? To repeat Gideon's question, if protein folds have been so stable, why have we had persistent problems in designing stable functional proteins? The answer to this is actually intuitively quite simple if you think about it. In order to encode function, you often have to encode buried charges, to locate buried charges. As you see here, this is a buried lysine, histidines. These are two divalent ions, metal ions, packed against one another. This is critical for function. They're buried atoms. They're not stable in that location. You need to accurately configure multiple functional groups. If this histidine moves out of the way, the metal ion won't go there. So you have to be very accurate. And all this accuracy, what it means, what it entails, is that you need networks of supporting interactions with the backbones of these loops. You have to encode hydrogen bonds and packing interactions with each of these residues. So, and throughout all this, you also need to encode protein stability, because below a certain threshold, as Gideon alluded to, the protein, maybe the active site would have formed if the protein were more stable, but it's just not going to form. You need a stable protein for function. So all these, when we were designing, just a second, when we were designing folds, we didn't have to worry about that. We only needed to worry about this last item on the list. Now that we're more interested in function, this becomes important. Yes?
When you search the PPD for protein, you never find it with the residues you're looking for on the list?
For hemagglutinin, for instance, absolutely not. And the natural proteins are completely inert towards hemagglutinin. Well, it's a combinatorially very difficult problem, right? If you're going to design 10 to 15 residues on the surface of a protein, the combinatorics of this are huge. And also keep in mind, this is very important. This again addresses Gideon's question. Think about these hemagglutinin binders. What we designed essentially are very hydrophobic surfaces. You're not going to see those in nature, right? On the very surface of a protein, such a stretch of phenylalanines, leucines, and methanines, this is going to be incredibly rare, very destabilizing. Right? So you're actually designing an unstable protein when you're designing function. And the question becomes, do you understand the compromise? Do you know when you've gone too far? You've destabilized the protein too far, stop here. That's a key question when you're designing function. So yeah, we've decided to focus on antibody design. We've recently expanded to enzyme design. I'll show you that as we move on. The reason for focusing on antibodies is that we have so many molecular structures of antibodies, more than 1,300 molecular structures of antibodies in the PDB. We understand antibodies much better than other protein families. They're inherently modular. I'll show you why this is important. And antibodies primarily use loops in order to interact with substrates. In fact, they use 6 mostly loop regions. They're called the complementarity determining regions, 3 on each of this is the heavy, the light chain, 6 essentially loop regions in order to interact with antigen. So if we're going to force ourselves to work with loops, the antibodies are a good way to focus our minds on that question. People have been designing or engineering antibodies for about 30 years. This is actually some of the most successful protein engineering examples come from antibodies. And the leading concept in the field, again, for 30 years and more, has been that antibodies are modular. We know that because this is how they come through our immune systems. And if you look at the structures, you also see the modularity. Usually there is a framework, which is quite constant, and there are CDRs sitting on top of this framework. And people suggest, starting with Sarel Fleishman and Greg Winter, suggested that you could take the CDRs from one antibody, transplant them on another antibody, and you'd get new functionality, or maybe the existing functionality from one antibody, transplant it to another. And this actually has worked quite successfully in the past. But what people noticed when they did this time and again, this is, by the way, the basis for antibody humanization. You find an antibody in mouse that binds your antigen of choice, and now you want to humanize the antibody. You take these CDRs and transplant them on human. But what people have found time and again when they did this experiment is that often the antibodies lost their stability when you did this transplantation, their affinity or specificity. The antibodies misbehaved. And then they start engineering the antibody. So there are solutions to these problems. But we want to be able to do this computationally. And one of the things, now I'll show you some of the lessons we've learned while trying to model and then to work with, experimentally work with the antibodies in our lab. So one of the lessons, the first lessons we've learned, took us a year, but still first, one of the important lessons we've learned is that while people have been traditionally thinking of the framework as this sort of conserved, constant region in the antibody variable domain, in fact, it's quite obvious, the framework forms important stabilizing interactions with the CDR. So this is shown here. You have one heavy chain here, another heavy chain here. Imagine you try to transplant H2 from this antibody to this antibody here. If your framework has a leucine or an arginine at this position, this H2 will not be able to form correctly on this framework. Right? So you can see here that positions in the framework, this arginine or leucine here, can control the configuration of the CDRs. Right? And if you don't know that, you'll design proteins which are unstable, unspecific, maybe non-functional completely. So this is one lesson, that you actually need to conserve key interactions between the framework and the loops that sit on top of it. And this is actually a lesson we can also transfer to Tyndalls and other proteins. Any questions? Another lesson that we've learned, and again, something that is known in the field, but you needed to sort of develop a methodology, computational methodology for this, is that antibody backbones, even conformations that have the same sequence length, can adopt different conformations. And those different conformations have different sequence propensities to them. So it's not enough to know that a particular position is an H3, it's a position 3 on H3, for instance. That's not enough in order to know what sort of sequences can occupy that position. You have to know which conformation it belongs to. So what we've done in our method is to cluster different conformations of, let's say, H3, find the sequences that fold into that conformation, generate these sequence probabilities for each individual amino acid residue, and then use these as sort of guides while we're doing design simulations. So when we design a new antibody, since we know what conformation a particular H3 belongs to, we can say whether some residues can fold into that conformation and others cannot. So for instance, if you look at the blue conformation here, at the third position there's typically an arginine, whereas in the yellow conformation there's an aromatic at this position. And these rules are important for stabilizing, for making sure that the conformation would be as you see in the PDB. Okay, so we have two sorts of rules. One of them is that the conformations of the scaffold, the framework, really are key to maintaining the conformation of the CDRs, and two, that there are particular sequence conformation rules that you need to maintain through design simulations for designing stable and conformationally stable proteins. And basically our strategy follows this sort of conformational search idea. We take two antibodies from the PDB, a gold and a cyan antibody, and we can design a chimera whereby we splice in the H3 from the gold antibody onto the framework plus CDRs 1 and 2 from the cyan antibody. And then we get this chimera. You obviously need to design the sequence in order to fit the new context of this antibody. And as we're designing the sequence, we're maintaining these sequence conformation rules that I showed you in the previous slide. And in this way, we can encode with the local and more global interactions that are important for the stability and function of the antibodies. Let's see this. Yeah, and the key point about this, the bottom line, is that if we have 1,000 antibodies in the protein data bank, and we can do this sort of conformational recombination on the computer, we have billions of scaffolds. Just by recombining different pieces from different fragments from different antibodies, we can generate billions of scaffolds, billions of different backbones for design of function. The entire PDB has about 100,000 protein structures, so this is orders of magnitude more structures than we've had, for instance, for working against hemagglutinin in the previous slides. Okay? So this is a movie. The algorithm development was done by mostly by Gideon Lapidot in the lab. We have a target molecule. This is the site we're targeting. We start by low resolution docking. Now you know what this means. Low resolution docking. You see that there are no details, no atoms on the protein. It's just some sort of a search through a more smooth energy landscape. We then refine the antibody, but when I say refinement, it's not exactly what I said before lunch. Here, by refinement, I mean really large conformational changes where we swap in pieces of the backbone of the antibody with other pieces taken from other antibodies. And when we swap a piece in and out, we design the sequence of that piece and actually also the surrounding environment, both for stability, for the antibody to be stable, and also for binding to the target antibody. So you can see that during the simulation, the antibody is sort of designed to wrap around the target site. Okay? The end result of this process are antibodies that are very different from natural antibodies. I had this question before, what if you take a natural fold and just design its sequence? You get a very different sequence. Here too, we get antibodies if you try to blast them against the human, mouse, rat, doesn't matter, germline. You see more than 50 substitution relative to any of the mammalian germlines. Although, if you look at them structurally, they do have the same types of interactions that you see in natural antibodies, including these very dense polar interaction networks between the ligand and the antibody, dense interaction networks to stabilize the conformation of the CDRs. These are canonical interactions that you see in natural antibodies, and also these high shape complementary interactions with the ligand, again because the antibody sort of wraps around the target. Here, Christopher Noone tried to see whether this algorithm does it make sense. So I mentioned in the previous talk that one way to see whether your energy function and conformational search are actually good enough is to see whether you can predict the conformation of natural proteins. And if you see a large energy gap between near native and far from native conformation, that's a good sign for the energy function and conformational search. So he did that with blind predictions, and as you can see here, he gets very close to the natural, the PDB structure for the antibody, including in the CDRs. So you can see here that even the rotamers are often recapitulated to high accuracy. And you can see down here that often we see fairly large energy gaps between near native and far from native conformations. So people have been doing this sort of modeling of antibody structures for many, many years, again, starting from the 1980s. But in all of these previous works, what people have done was to use homology as the guiding principle for choosing which CDRs to take, so very similar to homology modeling, which CDRs to take from which antibodies. So recombining the CDRs from different antibodies, the highest sequence identity CDRs, the highest sequence identity framework, and generating a model, and then maybe relaxing that model. What it turns out to be, what we found during doing these simulations, that often the highest sequence identity conformation is actually not the best template. We've seen cases where there's a 100% sequence identity template for H3, let's say, which is about 3 angstroms away from the natural, from actually what you see in the PDB, whereas there's maybe a 20% sequence identity conformation which fits exactly the conformation seen in the PDB. And by just choosing conformations based on energies, we can select these, the correct conformations, often, not always. This is an even more blind prediction where Debbie Fast from the Weizmann Institute gave us the sequence of an antibody she was going to, she was in the process of crystallizing before she crystallized it. Christopher ran the simulations, got a prediction, we sent it to Debbie, and then when she solved the structure, we got this. It's very close. So this gives us a good indication that, yeah, this conformational search algorithm and the Rosetta energy function are a good way to search and design antibodies. I should mention, maybe going back, since this is a more general principle here, that the loop structure prediction is a very difficult problem in structure prediction in general. And these are all the comprised loops. The key point here, if you just do loop structure prediction in antibodies, you don't see this high discrimination between near and far from native conformations, this high energy gap. What you normally see is very flat energy landscapes. The thing about this simulation is that we're choosing from a pre-existing menu of conformations. We're not actually, this is a discrete choice. It's a very large choice, combinatorially a very large choice of backbones. But we're not allowing just any conformation when we're sampling different conformations. It's just the conformations that exist within the PDB. Any questions? Okay. So now to antibody design. And here I'll start by explaining something about the experimental methodology, which also addresses the question of what is affinity maturation. And before that, just to give you some outline for this, we realized very quickly, and I'll show you the results, that we need to first understand how to design stable proteins before we design functional proteins. And this will be the outline for this part of the talk. So how do we do, what sort of experiments do we do? So for screening, in this project, we've already been screening more than 600 designed antibodies. To do this process effectively without, expressing and purifying each of the antibodies on its own, we're using yeast cell surface display, which is a method whereby if you, let me go forward, if you transform yeast with your designed binder, it's expressed on the yeast cell surface, It's fused to a C-terminal cymic tail, which you can detect with a fluorescent antibody. The target molecule, let's imagine insulin, maybe hemagglutinin, is also fluorescently tagged, so that if you incubate the system, you have two fluorescent signals, one for expression levels, and one, this guy, and one for binding, the red fluorophore. And now you just take this through facts, very, very simple. And on the x-axis, you report expression, y-axis is binding. Each of these individual points is an individual yeast cell subjected to 2 lasers. And this is the sort of plot you get where you have high affinity versus no affinity or no target. It's a very clear signal for binding. You can also use this in selections. If you introduce error-prone PCR libraries, you can very quickly select within really 3 or 4 weeks. You can select higher affinity binders using this strategy. So just keep in mind this fact that we can monitor the expression levels of our proteins. This is very important for the next slide. So at one point when we stopped, we thought we had a very good antibody design algorithm, and we tested it against insulin. We designed 17 potential insulin binders. And then we went to the lab and tested whether they would bind. But before that, we tested whether they express. And these are the expression levels on these 17 designed antibodies. And these are the expression level. This is the expression level of a highly expressed antibody on the surface of yeast. Now, for those of you who don't know yeast display, I'll just save you the trouble. These don't express. Essentially, even these 3 points here are 0 expression. The antibodies we designed were uniformly not expressed on the surface of yeast. So then, what is the reason for that? It's immediately clear when you see something like this that we're compromising the stability of the antibody. We were focused on this idea, similar to my previous hemagglutinin work. We were focused on this idea that we're designing binding sites, active sites, targeting a particular molecule, and we sort of forgot. We didn't completely forget, but we didn't realize how important it is to encode the correct interactions to make sure that the antibody is actually stable. Okay, so over the course of a year, we decided to resolve this problem, and we've been able to do that. So now uniformly, all the antibodies we design are uniformly well expressed, usually even better than this high expression benchmark. And how did we get from this to this? What we did when we saw these results, which were very distressing, what we did is everybody in the lab received copies of the models for all these 17 designs and some naturally occurring antibody structures. And we asked them, what do you see in these designs that is different from the naturally occurring antibodies? Tell us what you see in these designs that's different and important for stability. And we had a group meeting for about 2 hours where people complained about this arginine here. It doesn't make sense. There's voids between the light and heavy chains. There's so many problems with these antibodies. You're not stabilizing the loops correctly, not enough hydrogen bonds. I wrote down everything, and then everybody on the team, the antibody design team, we had on the board in my room, we had a list of all the complaints, what we think would be the solutions to these complaints, algorithmic solutions. And over the course of a year, we encoded all these ideas computationally to improve the algorithm. So you can see here, for instance, this arginine here doesn't have a countercharge. Here we had voids between the light and heavy chain. In the end, we reached a state where our designed antibodies don't appear to us using any structural parameters that we have to be any different from natural antibodies. And this shows itself very nicely in the experimental data on expression levels. What's important about this is that in design, we sometimes, not always, we sometimes have this sort of virtuous cycle where you have a hypothesis, which you encode as an algorithm. You go and generate designs using this algorithm. You then go to the bench, experiment. If it works, great. Usually it doesn't. So you refine your hypothesis. And then you have this cycle. Okay, and this you see within a very short time, a year. You can get from 0 to something quite useful. So when we got antibodies that actually express well, we also got binders. This is an example for a model. At this point, we don't have an experimental structure for this antibody that targets insulin. You can see, again, all these structural features that are important for binding, including high shape complementarity, charge complementarity, again, high shape complementarity, as you see here, These are some binding data showing that mutations that are supposed to disrupt the interaction with the insulin also disrupt binding levels. Mutations that are supposed to increase the electrostatic complementarity, insulin is very negative, show us some increase in binding affinity.
So did you discover one of the antibodies against insulin?
No, these are very different from natural antibodies. And there are no structures for natural insulin binders, antibody binders. We chose, yeah, we chose.
But there are antibodies against
Yeah,
insulin.
yeah,
so
yeah,
you
yeah.
can at least check the
We checked. There's no
homology.
no connection with any antibodies. So again, 50 mutations at least from the germline. And the affinity is about 50 to 100 nanomolar, both before and after affinity maturation. There's a change in what the affinity maturation process, we think, locks down the active conformation. Before we have structures, I'd rather not discuss exactly what the affinity maturation process does. We don't quite know. But essentially between 50 and 100 nanomolar using SPR. So this is purified antibody attached to an SPR chip and measured against insulin. So this is suggestive, or we're still again waiting for crystal structures and more characterization. We're now really turning to see whether the strategy of combinatorial backbone design, where we take backbone fragments from different proteins, homologous proteins, but different proteins, and you recombine them and then design the sequence in order to fit best this new conformation, whether we could extend that to other protein classes, and most importantly, tin barrels, which are also highly modular, not as modular perhaps as antibodies. They've been quite tough. And again, this is work by Gidon, and this time with Olga Chersonsky. And with tin barrels, the question as a model system for our work with tin barrels, this is also in collaboration with Danny Tufik from the Weizmann Institute. We've looked at homologous protein families. Some of them are phosphotiriesterases, which catalyze the cleavage of this bond here, and some of them are lactonases, which cleave this bond here. These molecules appear very different. For me, this is quite surprising. These molecules appear very different. The reaction they catalyze is quite similar, though. It's both of them are esterases, and geometrically they do it in a very similar way. So essentially, these protein families, even though they're reacting with very different molecules, they have some very highly conserved elements, including this is an overlay of phosphotriesterase and lactonase. You can see that many of the functional groups are essentially in the same spots. Again, very high accuracy for the catalytic sites, despite huge changes in the loop regions. So we thought this is a wonderful system to work with in order to understand what we can do in order to swap pieces in and out. Can we do that? Can we actually swap pieces in and out from lactonases and phosphotriesterases, generating new proteins, and would they have the necessary function? If they do, that's a very, very sensitive indicator for the location of each of the catalytic groups on the loops. So even seeing some weak function in these cases gives us an indication that we've done something right. We're getting the catalytic groups in the right geometry. Oh yeah, this is a movie. It runs for 8 hours. This is what Gideon tells me. I won't do this to you. I've never done this to myself. So again, you see this swapping in and out of a segment, redesign. I think this is a lactone. I'm not sure, but it minimizes while you're designing the protein. A lot of flickering back and forth. This is the process, and this is just for one blade. When we do this in actuality, it goes around most of the blades of the protein. And you're getting, again, similar to the case of antibodies, you're getting very different sequences from natural lactonases or phosphotriesterases, although these are many, many different designs. You can get thousands of different designs, all of which have the catalytic groups essentially in the same spot. But you can see that the loops are highly divergent. How does he do that? How does he get the catalytic sites to be in the same site, in the same place? What Gideon does essentially is say, I've seen how a lactonase reaction, what a lactonase reaction looks like in nature. There are about 10 residues. I'll fix them. I'll say the coordinates for the histidine need to be as they are in this lactonase. The lysine should be right there. He says exactly where all of these catalytic residues should be, including the active groups on these residues. And then he says, give me all the fragments, all the conformation fragments from the PDB for these two families, recombine them, make sure that the residues are right where I want them to be, and then redesign the rest. So he's essentially folding the protein, the timbrel, around the active site. Okay, this is the idea behind his computational method. Similar to the case of antibodies, what we've seen is that the first generation timbrels, design timbrels, did not express, or they were completely chaperone dependent. They were very, very hard to work with. But then with time, this is already iteration 4, they're now uniformly well expressed. They're actually very well expressed now. And in some cases, we actually see low levels of activity in many designs now. This is the design. This is an enzyme, a very weak enzyme. It seems exciting that the design is almost as active as a natural enzyme, but it's a very weak natural enzyme. It's not diffusion limited or anything of the sort. So our design is very weak at this point, or designs are very weak at this point, but this is a starting point. These results are from yesterday too. So it's a starting point, again, for in vitro evolution, or for studying the differences between active and inactive designs in order for us to further improve the algorithm.
What did you change
In the algorithm, or
Yeah, when you say that you slowly change
the algorithm itself? Yeah. So it's very similar to what we've learned with antibodies. Some of it is really, really trivial and detailed. For instance, what sort of constraints to apply when you're changing the backbone, how to generate the sequence alignments correctly. For antibodies, generating a sequence alignment is very trivial, right? Because you know the genomic segments for each antibody. Timbrels are much more divergent. They're actually not a homologous family. They're a superfamily. So it's much, much harder to generate alignments. And if you make errors, you suffer for it. So with time, we improved our accuracy in that. So it's essentially a refinement strategy where, again, you run your simulations, you try, go to and see what happens. The alignment, and also how you model the conformations. Also, there's more loop elements in timbrels than there are in antibodies. So it's just a more challenging fold, I think.
We're trying to catalyze exactly the same reaction that we are catalyzing?
Yes, we're starting from a template that does one thing, and we're moving to the other thing. As we're doing this, we're generating timbrels that are very different. They have, from natural ones, even the deletion and insertion pattern on our designed antibodies are different from natural. You can't align them to any of the naturally occurring structures. So, yes, it's...
The core structure is the same.
The core structure is the same, yes. We're conserving that.
It's been designed by nature.
Exactly, yes.
But so it's not that...
It's not de novo design, yes.
Yes, so if you change the substrate or the reaction you want to catalyze, you would have to change the core structure.
Not always. All these enzymes, I mean, many enzymes are actually promiscuous. Again, just lactonases and phosphoterastrases are promiscuous with regard to one another. I showed you this is a lactonase reacting against a phosphoterester. So there is inherent activity. And this is the way enzymes are, right? Because the core catalytic machinery can do more than one thing. The question is, how is molecular recognition around the core catalytic machinery, how that is placed. And that's where you get the selectivity of enzymes. And this is what we're trying to encode. Yeah, we're conserving the key catalytic machinery. We don't want to play around with that. Imagine in this case of phosphoteresterases and lactonase, you have these two metal ions. Coordination of two metal ions that are so tightly proximal to one another is very difficult. So we want to conserve that. But everything around that, the molecular recognition elements with the aliphatic or aromatic groups, that we can change. Yes. Yes. Exactly. Yeah. Oh, ongoing. No way. Okay. Yeah, actually, it's the last section, so we're good this time. So just going back a few slides where I showed you these results, where we're now getting uniformly well expressed antibodies, and now also uniformly well expressed enzymes. At this point, we asked ourselves, okay, so we must have learned something general about stability of proteins while maintaining functionality. Can we apply this to very poorly behaved natural proteins? Can we take a very poorly behaved natural protein and engineer it, design it, so that while it maintains its function, its original function, it will now behave better. It will increase solubility, increase its thermal stability. So you might ask, why would you want to increase the stability of proteins? Are natural proteins good enough? After all, they are, for instance, our proteins are stable enough, right? We're alive. If they were unstable, they wouldn't be. But it turns out that if many natural proteins are only marginally stable, they're stable enough within their natural contexts. But if you take them out of context, for instance, a human protein, the example I'll show you is human acetylcholinesterase. In our bodies, it functions very, very well. It's the fusion-limited enzyme. It functions very, very well. But people have been wanting to overexpress it for many years. I'll show you why. In bacteria, and you get no functional expression in bacteria. You take the same natural enzyme, you try to express it in bacteria, and no functional expression. So the reason is that, if you want to go into heterologous expression systems, overexpression, you often want to change the temperature, right? You're taking an enzyme that's only stable at about 30 degrees. Now you want to increase its thermal stability, non-neutral pH, blood serum. All these things are changes in the environment. You need to engineer the enzyme or protein for that. And this is the work of Dee Goldenzweig in the lab. All credit goes to her. It's an amazing project. And really we had a lot of support from experimental collaborators on this project. I'll talk about acetylcholinesterase. This was a collaboration with Danny Tufik, Moshe Goldschmidt, and Yoel Zussman at the Weizmann Institute. They have all the expertise for this enzyme, and it wouldn't have been possible without them, and also other collaborators. I'll show you, I'll focus particularly on acetylcholinesterase. We're not the first ones to worry about this question of stabilizing, mutating a protein such that it becomes more stable but still maintains its function. And already from the mid-1990s, people have used what's known as consensus design. You look at the sequence alignment of homologous proteins to the target protein, and you often observe, just as I showed you for antibodies, that at certain positions you're free to mutate. Let's say at this position, maybe almost anything goes. But at this position right here has to be a glycine. Maybe this glycine is important for function, maybe it's important for stability. You don't necessarily know, but you don't necessarily care. What you do is you look at your target protein, and wherever it diverges, wherever you see an identity that changes between your target protein and this set of homologs, you change it to the consensus. And by applying that strategy, this consensus design strategy, you can often both maintain function and improve stability and solubility. The problem with this strategy, overall stabilization strategies, an individual mutation makes a small contribution to stability, under 1 kilocalorie per mole, usually much lower than that to the stability of the protein. And that means that if you're going to work on a very poorly behaved protein, such as acetylcholinesterase, you need to introduce dozens of mutations. And that is traditionally quite difficult to do, because when you're combining dozens of weakly stabilizing mutations, there's a high chance that inadvertently you'd introduce a mutation that actually destabilizes or harms the function of your enzyme. And the accuracy of these consensus design or even computational modeling strategies is not so high as to allow you to combine dozens of mutations in one step. And this has been the challenge that we have faced. We want to have an algorithm that in one step designs dozens of substitutions into the enzyme, maintains the function, improves the stability, and lowers where you wouldn't need to test many different mutants, just maybe one. Acetylcholinesterase is an important enzyme in neurological synapses where it cleaves or hydrolyzes acetylcholine. It's also the target, because it's so important, it's the target of nerve gas agents such as sarin, soman, VX. Bad people use it. And people have wanted for many years to generate a more stable version of this enzyme so that you could overexpress it in bacteria, maybe use it as a prophylactic or therapeutic measure, countermeasure for nerve gas agents. But again, for 20 years, this has not been possible. So what is our strategy? What do we do that's different? We actually use a lot of the strategies that you already know about, for instance, sequence homologs. The first step we do is collect sequence homologs from of the target protein, And at each position, again, we see what sort of substitutions are allowed. Any substitution that's unlikely to appear according to the sequence alignment, we rule out. Okay, so we only allow these sort of the top ranking substitutions, but not just the best one. We allow a range of substitutions. This typically goes from just 1, if it's a fully conserved site, to about 8. 8 residues is the most variable site. Next, we let Rosetta know about these sequence choices, and Rosetta models each of these substitutions singly against the wild type conformation. So essentially a single site saturation mutagenesis strategy on the computer. Rosetta models each of these changes and measures whether this change on its own is predicted to improve the stability, again while maintaining function, or disrupt the stability. Those mutations that improve stability go on to the next stage, which is simple Rosetta combinatorial design. Okay, but limited to the space of substitutions that we got from step 2. Okay, so we reduce the allowed sequence space tremendously by going through a sequence alignment and then by computational mutation scanning. So now from this reduced space, we look for the best combination. I'll skip this for due to time. I'll just go straight to the results. This is the best variant we designed for acetylcholinesterase. It has 51 substitutions relative to the human variant. You can see that they're spread all over the place. They encode exactly the same things you would expect for stabilizing mutations. We actually know quite well the basis for stability in proteins, for instance, buried hydrogen bonds, surface polarity, improved surface polarity, improved helix capping interactions, rigidify loops, for instance, a serine to this proline mutation, and this is I really love this pair of mutations. The human enzyme has this huge hole here, and you can mutate, you can change 2 leucines to phenylalanine and tyrosine, and actually fills in the gap perfectly. It's hard to imagine that we survive with such a cavity. Anyway, the experimental results were very, I think, quite dramatic. This is the human enzyme expressed in bacteria. This is the lysate activity, normalized lysate activities for the protein expressed in bacteria. Human acetylcholinesterase, so these serve as sort of a proxy for expression levels. Human acetylcholinesterase and lysate is basically dead. All the 4 variants that Moshe Goldschmidt tested are about at least 100-fold better, more highly expressed than human. And one of them, this is this guy, is expressed to about almost 2,000-fold higher levels than the human variant in bacteria. This, it's a dramatic number, but maybe 2,000 times 0 is still 0. The levels are actually quite substantial. You get about 2 milligrams per liter culture. And they've actually been able, okay, so first of all, it's also much more thermostable. This is the human enzyme, and it's 20 degrees shifted to the right in all these variants. And they were actually able to crystallize. This would be the first crystal structure of an acetylcholinesterase that was produced in bacteria. And these are the active site residues, and they're under 0.2 angstrom RMSD from the wild type. So even though we introduced dozens of mutations on this, throughout the protein, we get very close to the to the natural acetylcholinesterase at the active site. The protein actually was tested for its function. It catalyzes acetylcholine at near diffusion limited. The worst variant here, I think it's this guy, is 3-fold lower than the human enzyme. This one is, you can't discriminate between this enzyme and the natural one. And it's also reactive against VX, all the nerve gas agents to the same extent. So functionally, it seems like the natural enzyme. In terms of stability, it's much better. I'll get to the end, and then if you have questions. This is another example. So we've done this now for about 4 enzymes and 2 binding proteins, if I'm not mistaken. In all cases, all the variants we tested, or our collaborators tested in this case, were better behaved than the natural enzymes. So next study, what she did was generate a web server, which we call PROS, the Protein Repair One-Stop Shop. And she just had it there, and then we got an email from a group at Georgia Tech where they had this beta propeller protein, unknown function, but involved in glaucoma. 100 non-synonymous mutations cause a certain type of hereditary glaucoma, and they thought that this is related to stability, but since it's of unknown function, you couldn't apply selection. They couldn't apply standard selection methods on this protein. So they asked us whether we could help them. We just gave them the web server, and then a few months later we got excited emails because what they saw, this is the natural OLF domain. And this is the one design they tested. So in the presence of calcium, it binds calcium, and in its absence, showing that even calcium binding is maintained. The design protein loses aggregates completely. It's completely aggregation free, and it's actually the most, according to them, it's the most stable OLF domain known to them. So this shows you that really it's fully automated, unsupervised. Anybody, essentially, if you give it a crystal structure, you'll get a variants that are quite likely to be better behaved. This just shows you some comparison between different variants for different proteins for acetylcholinesterase, a DNA methyltransferase, phosphotransferase, and cellulase enzyme. Just showing you some statistics on these different cases. So usually we introduce about 10% of the proteins mutated using our method. In some cases, there are many mutations in the core. 14 in the case of acetylcholinesterase. This is a very large number compared to previous design of stability cases, but others are not so high in the core. Some proteins have a huge change in their charges. For instance, PT goes from a PI of 7.2, which is near-neutral and therefore aggregation prone, to 4.5 by acidifying its surface. Many more acidic carboxylates on its surface, and in some cases you get prolines. So for each case, even though this is a fully automated method, for each case you see a different sort of molecular solution. So there is not a one-size-fits-all, and I think I recall that Tolstoy mentions that each unhappy protein family is unhappy in its own way. So just to summarize this, design of biomolecular function is not a solved problem. Unlike fold design, it's still a very significant challenge. What we've been doing, the progress we've been making, is towards using constraints from evolution and from the PDB in order to help us turn design of function into a more robust and reproducible technique. And we hope that in the future we'll be able to do something like what I showed you for stabilisation design, where you could actually just approach a web server, say this is the reaction I want, this is the substrate I want to catalyze, and there would be some magic happening on the computer and you'd get some sequences back. We're not there. So thank you all, and thank my group members, funding, and any questions? Yes.
Yeah, maybe one question because we are. Okay.
Yes. Please.
If you try to apply your approach to predict, for example, the sequence of enzyme that in thermophilic bacteria, you imagine that I give you the homologous in mesophilic, and ask you to predict the thermophilic.
Yes, so
does it work?
David Schreiber here is doing just that. So what he's seen is that if you look at mesophilic versus thermophilic bacteria, and use just the right alignments. For mesophilic bacteria, you only use the mesophilic alignment. For thermophilic bacteria, you only use the thermophilic alignment. What you see, if you apply to the mesophilic bacteria, the proteins, this algorithm, it's a weak signal, but it is significant. There is something there. You see that it goes more closely towards the thermophilic bacteria. It's not a huge signal, but there is something there. We're still working on that.
Okay, thank you for the extensive.